welcome to the fifth video on the chapter Biotechnology Principles and Processes. This video is about PCR or polymerized chain reaction. Why are we doing this? We need to have multiple copies of the gene of interest that we isolated from our organism, isn't it? So we need to make multiple copies. How to make multiple copies? For that, we are going to use this mechanism called as polymerized chain reaction. Into this PCR machine, we need to add certain things and then we need to understand what is happening inside this unit. All right. So, what all we need to add? We need to add something called as primers. What are primers? Primers are nothing but nucleotide chains, tiny nucleotide chains. All right. Tiny set of nucleotides of different lengths. Other than primer, what are we adding? We are adding a DNA polymerase enzyme, which is specific for this, called as TAC polymerase, TAQ polymerase. What is special about it? It has the ability to withstand high temperatures, which we will be understanding why we will be using that. All right. So, what happens in a PCR unit? Let's understand. So, what all did we add? We added primers, which are nothing but small stretches of nucleotides, small stretches of single-stranded nucleotides. You can see here in the diagram itself, here one primer, here another primer. So, we'll see what that means. All right. Okay. So, in PCR, what happens is step number one, we have our gene of interest like so. This is our gene of interest, which is a double-stranded stretch of DNA, which we need to make multiple copies. To make multiple copies, as you know, DNA replication should happen. How will it happen? First, the strand should unwind. From each strand, new copies should be made. Same thing here also is going to happen. But in our natural mechanism, all these are done by different enzymes. Here, we are going to use heat and separate these two strands. So when you apply heat, that is the first step in the PCR unit. First step called as it is called as denaturation using heat. Very high temperatures are applied. With this, the hydrogen bonds between the nitrogenous bases of the DNA breaks, resulting in two strands of DNA which are separated like so. Now we have two strands of DNA. Each strand is waiting to be replicated. How to start that replication? For that, we have this tiny set of nucleotides called as primers, which will go and bind to its complementary place. Wherever its sequence is complementary, it will go and bind. So, step number two, what is step number two? Annealing. Step is called as annealing. 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 So first is denaturation. Using heat, you're separating double-stranded DNA into two single-stranded DNA. Second step is annealing, where primers go and bind to the complementary stretch of the double-stranded DNA. This one, primer number one has bound here. Primer number two has bound here. The same way, throughout the stretch of this gene, there will be primers binding. Once that is done, which enzyme usually helps in polymerization? DNA polymerase, right? The same way here, DNA polymerase, a special type of DNA polymerase called as TAC polymerase, which is isolated from a particular organism, a bacteria called as Thermus aquaticus. So probably they'll ask you this question: what is speciality about TAC polymerase? This can withstand very high temperatures in the PCR unit, and it is extracted from a bacteria called as Thermus aquaticus. All right. So now we have two strands of DNA. To the both strands, primers have annealed or bound. After that, DNA polymerase, which is Taq polymerase here, starts adding new nucleotides here as well as here. Then what happens? So for each strand, a new strand of DNA is made. For each initial strand, a new strand of DNA is made. Once this is done, this first step repeats. That is, this DNA would go into separation using denaturation, then annealing of primer, then again DNA polymerase making the DNA, adding the nucleotides onto it and enlarging the DNA, resulting in a new molecule of DNA. Likewise, around 30 cycles are done, you get around 1 billion times amplified gene. So the first one tiny stretch of gene that you have added, you will get 1 billion times the amount that you added by the end of PCR. This is why during the pandemic, most of you would have done a PCR test. Why is this PCR test done? It was done to amplify the viral genome that was probably inside your throat or your nose. It was extracted. Whatever extraction was made had very less amount of viral genome, with which you cannot understand whether it is actually one type of virus or not. So, in order to understand that, that genome was made amplified and then made into many copies from which you can understand whether you are infected with the coronavirus. All right. I hope PCR is understood to you. Now, end of this process, what do you have? You have many copies of your gene of interest, which you will be putting it inside a host, which we will cover in the next video.